This video serves to outline the triple sugar iron agar slant, or what's called TSI. The triple sugar refers to glucose, sucrose, and lactose. Glucose is a monosaccharide, sucrose and lactose are both disaccharides. This is a differential medium, meaning that most organisms can grow on it, so it doesn't necessarily choose against a particular organism, but they will produce different color changes depending on their particular type of metabolism. So also, besides the three sugars that are present in the tube, there is ferrous sulfate that acts as the iron source, and that will enable a color change, which I'll talk about in a moment, if the organism possesses one of two enzymes. The other indicator in there is phenol red, and phenol red is an indicator that's commonly used to detect waste products that will result in a pH change. And so we're particularly looking for an acid production, so that means a drop in the pH, so below 7. And that will cause phenol red to turn yellow in the presence of an acid. So common results that we hear are the terms K or A, and it's some combination like A over A, K over A, and those stand for, the A is for acid and the K is for alkaline. Of course, we can't use A again, even though alkaline starts with an A. So we use A for acid and K for alkaline. So if the organism is able to grow, you can get a K over K result, which indicates that the organism does not ferment any of those three sugars that are present. So it lacks the enzyme sucrase, lactase, and it's also unable to digest glucose. And another possibility is what's called a K over A. So the top you'll see here is red, or a pinkish color, and the bottom you'll see here is yellow. It's critical for this that you do a fishtail on the top, and then hopefully you can kind of see the stab into what's called the butt of the medium. So even though this is a slant, there's enough medium poured across the bottom that you're able to actually stab it with a needle as well. And so in a K over A result, we say that glucose was digested. However, the organisms did not have the enzyme sucrase or lactase. The organism was not able to break down sucrose and lactose. What happens then is acid is produced from the glucose breakdown, but not enough acid because the other sugars were not broken down, that it didn't turn acidic or that yellow color all the way up the tube. And remember that that's phenol red that causes that color change. So if an acid is produced, the phenol red will turn red. And if we compare that to an uninoculated tube, here on the left, that is a tube that has that phenol red that hasn't experienced that color change from a pH change. So always make sure you have growth and a good stab there into the butt of the medium. This is, from here down is what we call the butt. From here up we call the slant. So another possibility is A over A with yellow all the way through. So that indicates that the organism has enzymes to break down glucose, and then it has either or and or sucrase and or lactase, so it's able to break down one or both of the remaining disaccharides. So what will happen then, there's enough waste product, enough acid produced that you'll get an acid change all the way up the slant as well as the butt. What's also interesting to take a note here is that you can get CO2 production, which is yet another piece of information that we can get from the TSI medium. And you may see that as a bubble here in the back. You might see that as bubbles or cracking here in the bottom. That indicates that CO2 is produced. You also might see, this is a really robust CO2 producer, so you not only see the bubble in the back, but it actually produces so much gas that it pushes the medium up the tube, so it traps the CO2 down there. So we would say that this one is A over A with CO2 gas production specifically. And then you can also get an A over A, but now you have a black in the butt. And the black is also from a gas production. It can be from the organism possessing the enzyme thiosulfate reductase, which will produce an H2S, which will react with that ferrous sulfate that was an ingredient, if you recall earlier, in the medium. and that will cause a black color to form. And we also associate organisms that can produce that black color with motility. And the nice thing about the black color is you can really see here that from the original stab line here in the back, the organism was able to move throughout the tube because the organism possesses one or more flagella there. The other enzyme that can produce this black color, and we don't know which is which, we just know that the organism has one of those two, is the enzyme cysteine desulfhydrase, which will also produce this end product here of pyruvate ammonia, but also that H2S, and again you'll have that reaction of the black butt. So we'll see this also duplicated in the Sims medium in another laboratory. That black color can indicate the presence of one of those two enzymes. So that particular one, again, is associated with motility. So just to review here, we can have a K over K result. I didn't show an A over K, but you can probably kind of picture what that would look like with a yellow slant on the top and then red in the bottom, but I don't have an example for that. But I have K over K shown here, then I have K over A. Then I have 3 A over A tubes. Again, these two on the far right, the two I'm holding up here, would have gas production. It would be specifically CO2 gas, so you could narrow it down to the gas type. And the one here in the middle has black in the butt, and so that would indicate H2S production. And again, it's going to be the enzyme cysteine desulfhydrase or thiosulfate reductase that can both cause that change to occur in there. And again, the black there is associated also with motility, so you can make that assumption based on the black here for our laboratory that that organism is also motile. Now, one of the things that we run into is when we have A over A results, I said that there was a possibility. We know that the organism is able to break down glucose, but we don't know if the organism has sucrase and or lactase. So we can run other media in conjunction with that that have just sucrose in them or just lactose in them to narrow it down. Is it one or both enzymes? So we can actually kind of team this test up with others to really characterize the enzymes that the organism possesses. And that is the TSI, or triple sugar iron agar slant.